Welcome to Brain Ponderings, your window into the amazing world of neuroscience research. I'm Mark Matson, the host. Today it's my great pleasure to talk with actually someone I've known for a long time and followed her work, even though my lab didn't do much on what exactly she's working on. It's Michal Schwartz. She's professor of neuroimmunology at the Weizmann Institute in Israel in Rehovot. She has made major fascinating discoveries on how different cells in the immune system kind of reside around the brain and they respond to environmental challenges uh, in health and in disease they play roles. And particularly, she's shown that these immune cells, they're essential for the normal health of the brain, things like even learning and memory, neurogenesis. And then in response to injury, they actually have good effects. For example, there's these kind of interactions between different components of the immune system such that one arm of the immune system can suppress another arm that can potentially be harmful. So anyway, these are kind of the general things she's working on. So welcome, Mahal.
Thank you, and thanks for the invitation.
Now, you grew up in Israel, and were either of your parents scientists, or why were you interested in science when you were young?
So it's true that I grew up in Israel. My father was survival of the Holocaust, so he was completely uneducated, officially uneducated. But when I was very young, 10 years old, I was in a group of children that visited the Weizmann Institute, children that love science. I visited the Weizmann Institute and without any background in science and without parents with science, I had a dream that that's the place that I want to be. And that was my
Wow.
dream. I didn't know exactly which field. I thought that I'll go to mathematics because this was my strengths
Hmm.
in high school. Then I did my bachelor degree at the Hebrew University in chemistry, physics, and mathematics. And then I moved to the Weizmann Institute to fulfill my dream. And I did a direct program of PhD in immunology. I shifted from basic science in mathematics, physics, and chemistry into immunology. And when I finished my PhD in immunology, I had a dream which was not correct, retrospectively was wrong, that somehow the two systems, the brain, the nervous system, and the immune system, has a similar regulation. Of course, they are not, but they talk to each other. And that's how I came to the field.
It's interesting. In a sense, you're kind of like, so myself, I was interested in biology from the beginning. My math was not great. And so I kind of went straight into, biology, chemistry, cell biology, and so on. But my son kind of followed your thing. He was very interested in physics. He was a physics major initially as undergrad. And then I don't know if you know Stan Prusner. He discovered prions. Um, he came and gave a talk at National Institute on Aging, where I was at. I invited him to give a talk and had a dinner, and my wife couldn't go, so my son went with me. And Stan talked my son out of continuing in physics because he
That's
said,
familiar.
yeah, because he said, well, you spend a decade developing some method or something in a big group of 100 people and so on and so on. And what's the direct implications for human welfare and so on? And so he suggested chemistry. And chemistry is really that's the basics for everything you do, I
Totally
do.
agree.
Yeah.
Yeah. So
Okay,
I
so
shifted from immunology to neuroscience as a postdoc. And then I realized the two systems talk to each other. And the major reason why I'm in the field in which I am now is because when I was a postdoc, I learned that the brain cannot tolerate the immune system. And I thought that this could not be correct.
Right.
And
Yeah,
that's our,
it didn't make sense, right? Because I've often thought kind of this general idea that, okay, the idea the immune system is just sitting there, like doing nothing until you're infected with a virus or bacteria, then it does something. Or until there's an injury, then it does something. But it doesn't really make sense, right? Why would you devote all these million, what, trillions of cells, and they're just sitting there doing nothing? That doesn't make sense, right?
so I think what drove me is even further that we know that immune cells are not just to protect us against viruses and bacteria, but they are the healing of the body, healing cell of the body.
Yeah.
In the context of the brain, the dogma has been for decades, since Paul Ehrlich
Mm.
discovery of the blood-brain barrier, that the brain cannot tolerate any immune activity. So it's completely isolated. And to me, it didn't make sense the tissue like the organ, like the brain, that is definitely irreplaceable. You can transplant many parts of the body, but the brain never. It didn't make sense to me that such a precious organ has given up the opportunity to be assisted by the immune system. That's how I started.
And can you start out by giving some background on just for, there, this is mainly neuroscience series of podcasts. So can you talk about just kind of the basics of the immune system? There's an innate arm, there's the adaptive
Yeah,
and different
yeah.
stuff.
So definitely. So there is the immune system inside the brain, which are the resident immune cells of the brain that are microglia,
Yeah.
and they are belonging to the myeloid lineage. Outside the brain, the normal, regular immune system, you have the innate and the adaptive immune system. The innate are the monocytes, which are all the factory machinery of the immune cells are coming from the bone marrow. And they are migrating to the lymphoid organ, they're migrating to the blood. So you have the innate immune cells, which is mainly the macrophages, the dendritic cells, and we have the adaptive immunity, which are helper T cells, cytotoxic T cell. And within the helper T cell, you have different type of helper T cell, the Th1, Th2, and regulatory T cell. So the immune system is very sophisticated system in the sense that there is a lot of control to make us, to allow us to respond when we need, and to keep it under strict control that there is no overwhelming immune response. So there are many checked and balanced. The best example is that when we had the COVID-19, we all were vaccinated, but we create memory cells to make sure that they will be on alert when you need, when the virus infect you. So that means that we have a system to be alert when you need it, but to keep it under strict control when you don't need it. So in the context of the brain, the belief was that the brain is completely has given up the opportunity to be assisted by the immune system. And my team was the first with the high penalty at the beginning, showing that the brain needs, brain, I mean, central nervous system, it's the brain and spinal cord and optic nerve. They need the immune system for their healing. And it was a big, big struggle because when I first showed it in a publication which was published in Nature Medicine 1998, everyone told me, you're absolutely wrong. And I was cited in a negative way.
Well, the reviewers of your manuscript, they signed off on it. So, and they were probably top people in the field.
And subsequently, we published a year later, less than a year later, another paper showing that also T cells are needed for brain repair, central nervous system repair. And this we recognize that these are T cells that recognize a central nervous system antigen. So they are autoimmune cells.
Ah,
We coined the idea of protective autoimmunity. So think about it, coming with saying macrophages are needed, and everyone's told me you are wrong.
right.
T cell, and not only T cell, but autoimmune T cell, which means autoimmune disease. So it doesn't make sense. It was a long, long journey. And it's so nice that now we have evidence, not we, the community has evidence from numerous work. Many of them were elegantly done by Jonathan Kipnis. This year, he demonstrated that if he isolated T cell from a site of injury, of spinal cord injury, these T cells are protective and recognize CNS antigens. So it's like a closed circle because we showed it, but we didn't, at that time, we didn't have the tools to show that these are indeed CNS specific.
Okay, and all right. So then, can you get into how do you study this? So, how do you manipulate the immune cells? You do a lot of work in mice, right? How do you manipulate the immune system so that you can establish, what immune cells are doing, what, where, and when, and how? I guess.
That's exactly what I'm writing now. When, where, and how.
Uh,
Okay, so we have to distinguish between the role of immune cells in healthy brain plasticity, the role of immune cells in acute injury, in aging, and in chronic neurodegenerative diseases.
yeah.
So I'll start with the acute injury because that's how we progressed. So in acute injury, at that time, we use mainly two models, animal model, mouse model of acute injury, optic nerve injury, and spinal cord injury. To demonstrate that immune cells are needed, we brought into the system extra macrophages when it comes to monocytes
Oh,
or extra T cells. So in the first work, we manipulated the macrophages. In the second work, we manipulate the T cell. So we were assuming, and we know, that spontaneous recovery from any injury, acute injury, is very limited. We are still struggling with it. If God forbid an anyhow is victim of spinal cord injury, the recovery is very poor.
yeah.
So we anticipated, based on understanding, that if we increase the number of T cells that will home to the site of the injury, the recovery will be better. And we demonstrated in two different stories. One we demonstrated with macrophages and the other with the T cell. But at that time, we didn't know what are the relationship between this type of cells. We just demonstrated macrophages are needed and T cells are needed, and we manipulated in many ways. Remember that the first work that we published was 27 years ago. When we started this journey, the tools were very limited. So we did
And
our
and
best.
and Mahal, how did you specifically did you manipulate? So the macrophages come from the bone marrow ultimately, and well, the T cells come from the thymus. That's what the T stands for. So how do you manipulate these?
So in the first work, with respect to the monocytes, we
Oh,
introduced macrophages into the lesion site.
I see.
This was in our early study. Subsequently, when we understood how they managed to get into the CNS, we didn't have to inject them directly. We manipulate the system in a different way. With respect to T cell, it's like an anecdote. I thought that if we did spinal cord injury and we passively transfer T cell that recognize either brain antigen, myelin antigen,
Ah,
or passively transfer T cell that don't recognize any CNS antigen. And we were surprised to see that those that recognize CNS antigen were protective and improve the recovery following optic nerve injury. And this was 1999 in Nature Medicine, and we call it protective autoimmunity. Then subsequently, we spent a lot of time to find out why CNS antigen. Can we do it by instead of passive transfer of T cell by active vaccination? How can we do it without inducing autoimmune disease? So to find the balance, what is the type of antigen that we need? So we progress, slowly we progress. And then at a certain stage, we understood that the T cell and the macrophages have relationship in the recovery.
Mm.
In other words, the T cell facilitate homing of macrophages to the brain. And then or to the spinal cord. And then we understood that following spinal cord injury, the macrophages that home to the site of injury can home either from the meninges adjacent to the lesion site or from the remote choroid plexus. And then we found the different phenotype of the macrophages from the leptomeninges and the other from the choroid plexus epithelium. In other words, if I can summarize it, the beauty is that we discover, against all odds, that recovery from CNS injury is according to the same regulation of healing of any other tissue. But the homing is more complicated, but it's the same chain of event. You have injury, you need macrophages to clean the site of injury, and these are phagocytic macrophages. Then you have a second line of macrophages that are resolving the inflammation, and then our T cell that further support. So it's a chain of immunological event which is well orchestrated in time and space.
So you said that so the choroid plexus is kind of lining the ventricles of the brain where the cerebral spinal fluid is.
Yeah,
And so there's immune cells that do they go through the cerebral spinal fluid down to the spinal cord? Is that how they're getting there?
a very good point. Up until these days, we don't know exactly
Hmm.
how they home. We do see them along the canal,
Ah,
but we don't have strong evidence. What we can see also when we inject monocytes into the ventricles and you do spinal cord injury, you can see that the monocytes are not homing to any place along the cord which is healthy, only a place where there is an injury,
yeah.
because it's attracted by chemokines that are expressed at the lesion site. So it's a fascinating process. And what we found, the major role of the macrophages that are needed are resolving macrophages. They are resolving the inflammation. But the beauty out of all of this story, that while we were doing this work, at that time, Jonathan, part of this work, Jonathan Kipnis was with me, and two other PhD students. The two of them are now professor in Israel, one at the Weizmann and one in the Technion. And one of them, I think you know, Yaniv Ziv. He's a neuroscientist.
Yeah, and but Ron, what's her name? Ron Harold?
Ron, yeah,
She
Noga.
didn't she go to Boston or
She went to Boston.
yeah.
Yeah, now she's in Technion, and
Uh,
Yaniv Ziv were in Stanford, and now it's a Weizmann Institute. So the three of them were
so
PhD.
for people who don't know that, I think my understanding is that Technion is kind of like the MIT of Israel,
Yeah,
right?
yeah.
Oh, I
So
had one other question. So in the spinal cord injury model, were you manipulating the immune cells before the injury, or can you manipulate after and improve recovery? Because obviously,
yeah, yeah,
yeah.
yeah. Okay, we did
Okay.
it. We demonstrated it. Very
Yeah.
nice. But the point was when we did all of this work, which was huge effort over many years, these three students, PhD students, were at the same time in my lab, Yonatan Kipnis, Yaniv Ziv, and Noga Ron Ariel.
Yeah, yeah.
And we're discussing a lot if the immune system is so needed to the CNS, there should be more fundamental role for the immune system in the relationship with the brain. Because evolution doesn't acquire us with tools to recover from injury. And definitely the injury spontaneous recovery in the CNS is very limited.
Yeah.
So we're thinking that there should be more fundamental role. And that's, in my view, the biggest transition in the field of neuroimmunology. This work that we published in 2006 in Nature Neuroscience, Yaniv Ziv was the first author. There was work which was similar, which we did earlier in 2004, and keep this with me. But I think that the turning point was this work when we took and we said, okay, if the immune system is needed for healthy brain, let's put mice in enriched environment. It was demonstrated that in enriched environment, there is neurogenesis, increased neurogenesis. And we decided to put immune compromised animal like skid mice. You know what skid
Yeah.
mice? Severe combined immune deficiency. So they have a deficiency in adaptive immunity. We put them in the same cage and we see no increased neurogenesis. You put healthy animal, one with intact adaptive immunity, one with impaired adaptive immunity, one benefit from the enriched environment, the other not. In other words, it was the first time that we discovered that T cells are needed to support healthy brain plasticity, neurogenesis. We'd
And
also
so in
test,
those experiments, you had the immune deficient mice. They don't respond to environmental enrichment by increasing neurogenesis. And then the immune deficient mice, you put in some immune cells and then recover
yeah.
yeah. And
So when
which cells, which type of cells?
T cell, mainly T cell,
Okay.
yeah.
These are
CD4.
yeah, yeah. CD4 memory helper.
It's CD4 helper T cell. So
Yeah.
this was just at that time just helper T cell without any previous education. Subsequently, we found that it depends on T cell that recognize CNS antigen, again protective autoimmunity. Because when we took mice that are overexpressing T cell receptor to brain antigen, um, they did well. When we took mice that are overexpressing TCR for ovalbumin, which is irrelevant, they were low. So the bottom line that for the first time we found that healthy, and we also tested for cognitive ability. We tested it in 2004 in a paper that we published in PNS when Ioni was the first author. And then in the Nature Neuroscience, we did a big work with skid mice, with wild-type mice, with different type of mice. And the bottom line was that we found that cognitive ability, neurogenesis were impaired in healthy animal with severe immune deficiency. So that opened the entire field because the question was, okay, if a healthy brain is dependent on the immune system, where this immune cell sit? Because in healthy brain, you hardly see any immune cells. So that's the beginning of our work and Kipnis' work. He started on meninges. We started in the choroid plexus.
Now with environmental enrichment, I don't know if you ever came across Henriette von Prague.
Yeah, of course.
Yeah, yeah.
I will, of course, I know her very well.
Yeah, she was she was an investigator and where I was and I actually
Very good.
recruited. But, she did these experiments where it suggests that the environmental enrichment, the main thing is the physical exercise
Yeah.
that's stimulating neurogenesis. Now, with the immune cells, is there a role for brain-derived neurotrophic factor, which has been strongly linked to neurogenesis?
Um, we tried to see, we didn't see
Didn't see.
any drama. We
Okay.
didn't see any drama. What we do say, we can say that the T cell, later on we found that they are sitting, these are memory T cells that are sitting in the meninges and in the choroid plexus, and they affect the brain remotely via cytokine.
And
Subsequently, we found that also the T cells support ability to cope with stress, mental stress, and other people showed that kidneys show that they are involved in social behavior. So the T cell, the bottom line, the T cell now we know, and these are CD4 T cell, are supporting healthy brain plasticity, mental plasticity, social behavior, cognitive ability, neurogenesis, you name it. So this was very exciting, and this opened for us to the question where they are doing it and how they are doing it, and this brought us to aging and to Alzheimer. And
has anyone looked, we did a lot of work on intermittent fasting, right, over the many decades and found a lot of beneficial effects on the brain, including effects on neurogenesis and memory and injury responses. And so it would be interesting to look for you, since you still have a lab, I'm retired, to see how intermittent fasting, if that will enhance these beneficial effects of the T cells.
you mean fasting?
Yeah,
We did the other way around. We published in 2023, I believe, a paper where we gave the animal high-fat diet.
yeah,
And we found that it worsening cognitive ability.
yeah,
And this was mediated again by the immune system. We saw elevation of regulatory T cell. We found also metabolites that are going up that interfere with cognition. So definitely nutrition and food consumption, regulation, habits in food consumption as key role in affecting the immune system and in turn also cognitive ability. But we never did the fasting.
yeah.
I'm
There's
not
a
sure that
lot
we
of
would,
interesting evidence. So there's a lot of interesting relationships between energy metabolism and the immune system. And one thing that happens with the fasting is the cells switch from using glucose to using ketones as
yeah,
energy. And the ketones, at least one of them, has very important signaling functions. It activates transcription factors. There's a lot of evidence now that, for example, we showed that beta hydroxybutyrate, which is a ketone, that it will stimulate BDNF production. Another lab showed the same thing. And so I think it would be interesting to look at effects of ketone.
definitely yes. I'm not sure that we will get approval for fasting the animals, but
Oh, you know what? So when we started this work actually before I went to NI, when I was at University of Kentucky, so we have animal care and use committee, right?
yeah,
And uh, it had already been shown by another lab that every other day food deprivation extends lifespan in mice.
I remember, yeah.
Yeah, and so that ACUC was they were concerned, well, does this have an ad? But I said, look, I said everyone else needs to justify why they don't do intermittent fasting because it improves the, see what I mean?
Yeah, I know.
The normal conditions are unhealthy. The animals, they eat as much as they want. They don't get any exercise. So but anyway, you would have no problem getting it
No,
approved.
I was kidding, but
Yeah,
definitely I agree with you that the nutrition affect the brain
yeah,
and to a large extent via the immune system and not necessarily via the metabolites. The metabolites affect the immune system and in turn affect the brain.
yeah,
But for us, it was a turning point because we were desperately trying to see what's the underlying mechanism if the in healthy brain, which it's in healthy brain you hardly see T cell, You need them for supporting healthy brain plasticity. And this was also a turning point in our research on aging, because when we discovered this and we found immune cells in the choroid plexus, um, I thought that we need to understand whether aging of the brain is affected by the immune system, affected by the aging of the choroid, or by the brain. So we did robust RNA sequencing of which was at that time we were among the first. And luckily, I approached a scientist who came back from his postdoc to Israel, Ido Amit. So I collaborated with him and I told him that that's what I want to do. So we took 11 tissue from young animal and 11 tissue from old animal. And from both the young and the old, we took the choroid plexus separately from the immune system and from the brain. And to our surprise, it was so striking. They found a strong signature of type 1 interferon in the choroid plexus. Initially, when I saw it, I said, wow, my colony here at the Weizmann is contaminated with virus because type 1
Huh.
interferon, antiviral response. And it turned out that it's not. This is a response. We did in vitro and in vivo. This is a response of the choroid plexus to inflammatory cytokine that comes from the brain.
Ah.
Uh, we
Or like TNF or something like that.
Exactly,
Yeah.
exactly. So since didn't get approval to do parabiosis mice to connect and to see whether the aging of the choroid comes from the blood or from periphery, the student at that time, one of them, she's now a professor in Pasteur, she went to Tony Weiss to do the parabiosis mice, and we found that first, that is his mice also, the type 1 interferon in the choroid is going up in aging. Secondly, we found that indeed the signature is induced by the brain through the CSF on the choroid plexus. And when we neutralize the type 1 interferon receptor on the choroid plexus, we rescue the mice, we reduce cognitive loss. The beauty is that lately we found that in all neurodegenerative diseases, there is a strong signature of type 1 interferon in the choroid plexus, which impaired microglial function, which in my
Hmm.
view, it's amazing because think about viral infection. So maybe the down the negative impact on virus on cognitive ability is not because the virus entered to the brain, but because the choroid plexus protect the brain by upregulating type 1 interferon that keep the virus outside the brain, but negative, if it's chronically expressed, affect the brain.
Ah, it would be interesting to see what happens in long COVID. Uh,
So it was tested. There was a publication from the group in Stanford. They had access to brain of individual that passed away following a late COVID with cognitive impairment, and they found exactly this signature. So for me, our paper was published in Science in 2014. But later on, we found that microglial signature in aging is type 1 interferon. And on microglia, it downregulates transcription factor like MEF2C that help microglia to cope with inflammation. And then later on, a group in Mass General Hospital, published similar work in Alzheimer, downregulation of this. So the beauty of science that the dynamic, if you read the science, it takes you to the next step.
yeah. Okay. And okay, so with aging, you get this shift in the phenotype of these choroid plexus cells, and then you can, in mice, you can do manipulation to kind of prevent or reverse that age-related change, and then you get beneficial effects of that manipulation in models of age-related neurodegenerative disorders. Is that
Yeah.
right? And okay, then can you go in a little more detail on your work in the Alzheimer's
Oh, we should.
mouse model? Yeah.
It's one of my favorite topic. So because of the aging, when we're realizing that the aging of the brain to a large extent is affecting by the aging of the immune system, and since we all know that the key the major risk factor of Alzheimer's is aging, because 90% of the patient are age-related and only 5 to 10% are genetic.
Yeah.
So we thought that if aging is the major risk factor, and if we know that aging of the immune system impact the brain, we went into the Alzheimer. And our first work in Alzheimer, we took one of the mouse model that is very often used in research. It's the 5X FAD. It's the mouse model with 5 human mutation, 3 in APP and 2 presenilin. So this mouse model recapitulate most of the features that you have in Alzheimer patient. You see there first amyloid plaques, then you see, and with a long delay, you see cognitive loss. And now we know that even in patients, the disease is dormant in many years. When it becomes symptomatic, it's long after the disease was for many years asymptomatic. So to me, it like ring the bell that maybe the switch from asymptomatic to symptomatic relates to the immune system. And now we know that with the approved drug for Alzheimer's disease, the recently approved drug, that, yeah, okay. I'm saying that the approved drug. I
Right.
didn't say, but it's clear that they are very effective in removing the plaques, and there is very modest or slim effect on cognitive ability, which makes sense in now what we know, that by the time that the disease becomes symptomatic, it's after a long time that plaques were accumulated.
Yeah.
And my belief is that at this stage, the major drive of cognitive loss is the local brain inflammation. So taking together all of this led me to think that if we boost the immune system, we may be able to recruit macrophages and regulatory T cell that we knew from our acute injury model that are needed for repair. So that's what motivated us. And it
And
did,
Michal, were you able to get any postmortem choroid plexus from Alzheimer's and age-matched control?
yeah, we did it. We did it, and we found the same signature of type 1
Okay.
interferon.
Yeah.
In our paper in 2014, the science paper, we showed it,
Yeah.
that in aged people, and there is type 1 signature. And now that we understand more this type 1 signature, we published last year a paper where we show that if you take the published data about Huntington, frontal temporal dementia, uh, aging, and AD, and you analyze the data explicitly for the choroid plexus, you see the type 1 signature. So it's in all the diseases.
Okay, so then what about from the standpoint of translation to
I'm getting into
oh,
it.
okay. Okay.
So based on the understanding that we need to boost the immune system, we did a lot of work in the translation, and it occurred to me, and I'm saying to me because it was like jumping out of to go to checkpoint. Why checkpoint? Immune checkpoint. Because immune checkpoint keep the immune system under regulation. And there are inhibitory checkpoint, the level of which increase when the immune system is more and more exhausted. So among the family of checkpoint are the PD-1, PD-L1, TIM3, CTLA-4. PD-1 is programmed death protein 1, the ligand is PD-L1 and PD-L2. To make a long story short, when I thought that I need to boost the immune system, I said, let's block a little bit the inhibitory. It's like when you start the car, it's whether you push more the gas or reduce the brakes. So I think let's reduce the brakes and see if we can do it. And we checked several checkpoints, blocking several inhibitory checkpoints. And we ended up, the first publication was in Nature Medicine 2016. But then I decided that we need to check several checkpoints. And we ended up working with antibody to PD-L1. So using antibody to PD-L1, we managed to show that we can boost the immune system modestly. And we found initially in 2 animal models, one Alzheimer, the 5X1, and model of tauopathy. Because we activate the immune system, so we thought it doesn't matter which model we will use, any model that there is accumulation of misfolded protein, there is local inflammation and cognitive loss. And that's what we did.
So is this the memory T cells that you're
Yeah,
releasing
memory Ts.
you're releasing from inhibition by this
Check.
PD-1
Yeah,
mechanism?
PD-L1. Yeah,
And is this the same kind of thing that's being used in
cancer.
cancer?
Yeah. The initial yes, and then we ended up not because we used the antibody that exist for research, which is the first same one for cancer and for any other uses. Then uh, by deciphering the mechanism of action, we realized that the activity is driven by Cmax and not by exposure. That means that you have to reach a high dose to trigger the immune system, but exposure to the antibody thereafter is not needed
No.
because the antibody is needed only for the activation of the immune system.
Oh,
So based on the understanding of the mechanism, we engineered new antibody that allow us to reach Cmax without endangering with long exposure.
I see.
So we shortened the half-life of the antibody. So we did a lot of the mechanism of action. We found that it's macrophages and Treg dependent. So indeed, it's triggering an immune response, and with a delay, we see macrophages and T cell in the brain. And we found that in all animal models that we tested and others subsequently tested, we found improvement of cognitive performance and reduction of inflammation. But again, when I came out with this idea, again, there was a lot of skepticism. So it's difficult to be pioneer. I can tell you, difficult.
And
And this is
right,
the
and
story of
so
my life.
so you developed this new antibody. So, like, if you were going to do a clinical trial,
We
then
are, we
you'd
are
have
in
to go. You have to go back
so
to
we are
the.
in clinical trial.
Ah,
Yeah. And that's what I'm saying. The translation is coming.
okay.
So we founded the company in 2016. The company engineered this new antibody. It went through all the regulation. It went through tox study in monkeys. We got approval to go for clinical a clinical trial in UK, in Amsterdam, and in Tel Aviv. And we had to enroll 40 patients. We finished the enrollment a month ago. The primary endpoint is safety, and we are collecting markers for the proof of mechanism because
Ah,
it's mainly dampening the immune response. And then, of course, a marker of disease progression, but it's small number of patients in each cohort because we had to do because it's safety, we had to do 5 escalating cohorts of patients. But we are, I think in 2 to 3 months, we'll have the first hint after single injection, and in a year from now, we'll have the result of repeated injection.
That's very exciting. Uh,
It's indeed.
right. Do would you expect to see any changes in levels of some cytokine or some marker in the cerebrospinal fluid?
Yeah, because based on the mechanism of action, our primary effect is on the level of inflammation. So
Okay.
for the
Yeah.
mice study, we saw effect on GFAP, but this is in the parenchyma.
Hmm.
We found reduction of chemokine, which means that there was recruitment of immune cells to this chemokine. So we expect to see effect in the CSF on inflammatory cytokine or chemokine or GFAP. That's what we expect.
Now, you mentioned that the switching of the choroid plexus T cells during aging to this Th2, and then that's associated with increased, what did you say, interferon or?
So there are two, I didn't explain it, I didn't spend enough time on it. We see the choroid plexus in aging, a strong signature of type 1 interferon. So it's not interferon gamma, it's interferon alpha or interferon beta, but not the gamma. The gamma is
Oh.
the good one.
Okay, and then would
It,
interferon gamma be a treatment for
Uh,
Alzheimer's?
there was a work that was done after our work. They tried to do with interferon gamma and they found a beneficial effect. I don't like to go with any treatment based on a single cytokine for two major reasons. One, the story is not a single cytokine. It's a big operation. Secondly, when you go with single cytokine, you always have the risk of
So,
over, under. So
and you've got all these other organs that are being exposed
yeah,
that,
yeah, yeah. So my approach is always, but the beauty of our treatment that in 2022 there was a publication in Nature, not by my team, different team, showing that the same approach that we took, they call it anti-aging because it allow they demonstrated reduction of senescent cells in many organ tissue, in many tissue in the body. So they call it anti-aging. So I believe that you cannot treat with single cell, you cannot treat with single cytokine or single molecule. It's an operation.
yeah, well, exercise, physical and mental exercise, don't overeat, maybe do some intermittent fasting. These are things that we evolved
Of
with.
course.
Yeah.
Yeah. Yeah.
I did have a question about there's a lot of interest in the gut now for so many
Yeah.
reasons, right? The microbiota, the emerging evidence, there's strong communication with the brain, the whole story on the in Parkinson, where apparently the problem first begins in the gut and maybe goes up the vagus nerve.
Yeah.
So, what about uh the yeah, can you do you have some thoughts on the gut-brain connection in relation to your work?
Oh, yes. We had some work collaboration, but didn't reach completion yet. Definitely the guts. Um, there are several reasons of this communication. First, the metabolites are inducing the
Uh,
immune system.
uh,
So whatever the guts produce, some of the metabolites can induce the regulatory T cell, effector T cell. So the guts definitely. There are some cytokine that are produced by the guts that can be detrimental to the brain, such as Th17. So definitely the gut has a major role in controlling the immune system or the immune system control the gut, whatever you want, but definitely.
yeah. Yeah, these metabolites of the bacteria, like the short chain fatty acids,
Exactly. Yeah,
butyrate, there's a lot of evidence.
yeah, yeah.
Okay, so probiotics.
Probiotics.
Yeah. Let's see. Development. We didn't really talk at all about development, but and for people who know about the immunology, they know probably where these cells initially come from. But can you talk a little bit about, is there a normal roles for the immune system in the proper development and wiring and establishment of connections in the developing brain?
So definitely, but I'll start with a very nice anecdote related to pregnancy. So even before embryos, when we found that the neurogenesis is affected by the immune system and cognition is of a normal, healthy brain is affected by the immune system, one of my students at that time was pregnant. And she asked me if what we are finding relates to the fact that during pregnancy, we have some, very often women have problem with focusing
Yeah.
or with memory. I said, yes, it's common. And she asked me if I think it's connected. I said, of course it's connected because during pregnancy, the adaptive immunity is reduced
Hmm.
to avoid rejection of the fetus
Yeah.
because 50% of the fetus come from the partner.
Yeah. Yeah.
So we took mice, uh, wild-type mice, and skid mice. And we found that during pregnancy, there is reduction in neurogenesis in the wild-type mice,
Hmm.
but it's back normal after labor, after giving birth. In skid mice, there was no difference between the normal level and the pregnancy. So it tells you that the brain is affected by the immune system even during pregnancy.
Yeah.
Now, with respect to effect of the immune system on development, there is a lot. There is a lot. We did one single work, but there is a lot showing that infection during pregnancy can negatively affect the fetus,
Yeah.
and there are evidence related to schizophrenia. But I think that as of now, it's preconditioning. You need another trigger that it will develop or not. That's the common wisdom now. And again, it's a type 1 interferon dependent.
Yeah, and isn't there evidence that infection, inflammation, and also obesity and diabetes during pregnancy increase the chance of the child having autism?
This I don't know from my own
Yeah,
experience what you're reading, I'm reading, but I don't
I
know.
think that's what I've read. Actually, I wrote a little article on this with a student, and you can actually in mice there are some genetic causes of autism, and some of the genes seem to focus on this mTOR pathway, which promotes cell growth. And so the idea, at least with obesity, is if you're like have excessive chronic energy intake that and protein, you get excessive activation of the cell growth pathway, and you get kind of the brain is growing too fast. The neurogenesis are occurring too fast, and connections are forming too fast, and so the wiring gets messed up in ways that lead to problems.
Yeah.
Yeah.
Yeah, so I'm familiar with it and we did some work, but again, I think that it's the not the virus itself, but the mother response to the virus. This is my view these
Ah,
days.
interesting. Yeah. Huh. Oh, yeah, there's ways that could be tested and
Yeah, we
yeah,
tested it. We tested it.
yeah. But in an autism model, it would be interesting too to test that.
Yeah, yeah. But my hands are full and I cannot do more than I'm doing. I have more idea than I can test.
That's the way it always is, isn't it?
Yeah.
But that's a good situation.
Yeah, I'm still very much excited about the science. I'm waiting restless to see the results of the clinical trial
Yeah.
because, it's like going through 28 years journey from beginning till clinical trial.
Yeah, but it's great. There's not a lot of scientists that are able to do that.
Yeah,
Yeah. Fantastic. Okay, you're doing great. Keep on pushing.
I'm,
And what keeps you going is the students and postdocs.
yeah, what kept
Right.
me more doing is my motivation,
Yeah, yeah.
curiosity and motivation.
Yeah. Yeah.
Really,
And that's contagious.
that's contagious. Yeah. And I think out of my lab, there are already 16 or 17 professors all over the world.
Yeah,
So,
that's
which is
fantastic.
nice. And they are doing very well.
Yeah.
So,
Yeah. That's great. Well, in an hour we went through this quickly. What I'm going to do is put links in the description section to Dr. Schwartz's. She has so many articles, but she has some really nice review articles. She had one in Science a few years ago, fantastic review article with a lot of illustrations, one in Neuron recently. So for those of you who, just by the nature of the topic, it's very complicated. You're talking about the immune system and the brain. Um, but it's fascinating work, and if people want to follow up, they can find those easily, or just Google Dr. Schwartz. She's easy to find. Put her name on PubMed, and you'll find her articles. Okay, thanks a lot, Mahal.
my pleasure. And take care and stay well.
Okay, you too.
Bye bye. Bye.
Bye.